Jai Hind everyone. Hope so you people are doing well. And today lecture is about the glyoxylate cycle. First of all, I want to share one information that we are going to conduct a IIT JAM GATE and GAT-B live classes. If you want to join there, the registration form link is available in the description box. You can go and register. The registration charges will be just 100 rupees and you can avail a demo class. If you like that, then you can continue for the by just by paying the course fee. So let's begin glyoxylate cycle. So glyoxylate cycle is something which is not present in the mammals, but the intermediate products are very important in the all of the organism. Either it is a mammal or it is a vertebrate, it is a anything. So glyoxylate cycle, if it is not present in the mammal, then where it is present? There is an organ which is called organelle which is called a paroxysm. There is one organelle which is called what? Paroxysm. Okay. This paroxysm is modified and do the able to perform a catabolism for the acetyl coenzyme. Acetyl coenzyme. So this acetyl coenzyme A can be converted into the glyoxylate and the succinate. That cycle is called as a glyoxysm. So instead of doing the its own function, it is going to do a little bit of different function to get energy source from the non-carbohydrates like amino acid, like fatty acid, like purines, nucleotide. So if you are getting this type of things, which is purine, which is protein, which is fatty acid, if you are getting energy from such sources, then in case of the plant, protozoa and the fungi, this is happening in the glyoxysm. Glyoxysm. Now you understood glyoxysm. Why? The same it is a vesicle which is called as a paroxysm. The same vesicle now it is called as a glyoxysm. So why it is called as a glyoxysm? Any guess? Of course, correct. If you are saying glyoxylate is forming, then it is correct. It contain a glyoxylate which is in which is happening in the glyoxysm vesicle. So it is called as a glyoxysm. Now this is the important intermediate which is present in the which connecting the glycolysis, then TCA cycle, tricarboxylic acid cycle, and then furthermore, this glyoxylate cycle is first discovered by the Korenberg and the Kreb. Today's lecture we will discuss about first thing we'll completely discuss the glyoxylate cycle, glyoxylate cycle. Then we'll discuss about the important current research going behind this glyoxylate cycle for now. So let's see. Glyoxylate cycle was first discovered by Korenberg, Berg and Grape. I will suggest you to fast forward this video so you can have a better experience of this video, because while writing it will be taking time. So you can choose a speed according to your understanding. So it is discovered by the Korenberg and the Grape. Do you know the another name, another discovery which is done by the Korenberg? If you know, please do the comment. I am going to give you the most important thing to that person who is going to give the correct answer. Just by you will get one coupon in which you will get a discount. at least. Okay. So Korenberg is uh, known for the one more thing, which is you have to tell, and then Krebs cycle, which is also giving one more important cycle, that is called TCA cycle. Correct? That is called a TCA cycle. Let's see. So we have a different sources. Let's see what are these. So my dear, there is something I written wrong. That is a spelling. Glyoxysome. Okay. Sorry, peroxisome. So this is the three important ways by which the glyoxylate can be formed. So you must have known the purine degradation. So in purine degradation, the purine will be degraded into allantoin, and then uridoglyocolate hydrolase will be converting the uridoglyocolate into the glyoxylate. Another thing is the fatty acid degradation by which beta oxidation. Note it down. By the beta oxidation, it is giving the acetyl coenzyme A, and acetate or activation of ethanol oxidation will give you the acetyl coenzyme A, which is converted into the isocetrate. Isocetrate lyase, which is very important enzyme for glyoxylate cycle. You remember it, okay? Isocetrate lyase, isocetrate lyase is converting isocetrate into glyoxylate, and then glycolate glycolate oxidase, glycolate oxidase producing what? Producing the glyoxylate, and then glyoxylate is going to give you the malate by the help of enzyme uh, malate synthesis, and then glycine will be formed, which is an amino acid. That is that is done by the enzyme glutamine glyoxylate aminotransferase. So this is the whole theory, and the photorespiration is also important. The photorespiration is occurring in the plant where the RUBP is not going to bind with the CO2. Instead of binding with the CO2, it is binding with the oxygen, and it is going to give you the 3-phosphoglyceric acid, as you remember, and I am going to show you that also, and plus glycolate, 2-phosphoglycolate. So this glycolate is converted again into glyoxylate. So these are the important thing which is we are going to cover. Now this is the very important cycle which you have to remember. Okay, and let me highlight those important enzymes which is important for the glyoxylate cycle. So these all are the precursor for the glyoxylate. Now we'll start from this point. Okay, at this point. So glyoxyloacetate is binding with the acetyl coenzyme A, and this acetyl coenzyme A is converted into citrate. Acetyl coenzyme A and oxaloacetate or oxalate will bind and give you the citrate. And this CIT is what? CIT is citrate synthesis. Okay, I will write separately here some in this enzymes full form. Okay, don't worry about it. Now citrate is converted into isocitrate by the aconitase enzyme, aconitase, and then isocitrate is break down into the glyoxylate and succinate, which is going to succinate will go for the TCA cycle, and from TCA it can go for the intermediate of the TCA, then will go for the uh, reverse of glycolysis. Okay, it will go for the then glycolysis reverse. Why I'm saying reverse, you can write better that glucose synthesis, glucose synthesis. So if you don't know how this is happening, you can watch our own video in our channel, which is given that how glucose will be gluconeogenesis is occurring and how this glucose is synthesized. Now, now the thing is that photorespiration. If you'll see photorespiration, RUBP, ribulose 1,5 bisphosphate in presence of Rubisco enzyme, it is binding with the oxygen and it is going to give you the 2-phosphoglycolate, 2 molecule, and then uh, it will convert it into the glycolate, and this numbers of cycle, numbers of the steps, then finally it is going to give you the 3-phosphoglycerate and 2-phosphoglycolate. Okay, so these are the important product which is synthesized. Which amino acid which is intermediate in this? These are the glutamate and glutamine intermediates amino acids are there. So finally you will get the glycolate from this cycle as well. Now another is the purine degradation. In purine degradation, it is converted into the 5-hydroxy isocetrate. Then it is converted into the allantoin in presence of H2O, and here the one peroxide is synthesized. Allantoin is converted into the allantotate, and then it is converted into uridoglycine. Uridoglycine is converted into uridoglycolate, and then presence of uridoglycolate hydrolase enzyme, it is converted into hydroxyglycine, and then finally glyoxylate. So there are a lot of enzymes which is written here. You don't worry about it because here we are not going to see very deep into that. If you are going to go for the particular research, then you can search and you can ask me. I will give you the all details. But the enzymes which is mentioned here in this, we will write all the enzymes name separately here. The question comes here in your mind: in which organelle or in which space this all things is happening? Can you answer me? Anybody, please, in chat. So these are the important enzymes which is written here, the all the full form, and at least you should remember the enzymes which are present inside the glyoxylate cycle. Okay, this is your glyoxylate cycle. Oxalate cycle. Okay, so you must remember all the enzymes of the glyoxylate cycle, but you can remember all the other enzymes which is given here over for the precursor. Now, I ask at which place these things is happening. These all things is happening in which organelle. So it is, of course, it is present in the plant cell, plant cell, and fungi, some fungi, some protist, some protozoa or protist. So there is one protozoa, maybe some is small protozoa. Definitely in all plants, but here you can see these are the in some organism. Okay, not in everyone. So if you'll see the plant, plant is a major site for the glyoxylate cycle, specifically in the seed germination. In what? In seed germination. So seed germination having mostly fat. It store fat. We can say like oil, and this oil will be having TGA triglyceric acid, and this triglyceric acid is converted into the acetyl coenzyme, and this acetyl coenzyme again bind with the oxaloacetic acid, and then it will go for the further steps which we seen here. So usually the concept, this which we seen here, the concept is that this acetyl coenzyme is a 2 carbon compound. So usually the 2 carbon compound is not used for the source of energy, but here in the germinating seed, the 2 carbon compound as a acetyl coenzyme may use for the carbon source, like for the source of energy and for the germination of the seed. So that is an important thing which is rare in other organism, like in mammals, and we don't have such kind of pathway. Now, the other than the acetyl coenzyme, you can see citrate, sorry, acetate is used for the carbon sources. Now, what we will do after reading this, why we are reading these all things, how it is going to help us. So, if you see the current research, which I am going to discuss now, then you will understand why it is important. So, before I will start telling you what are the important things, why we are reading this, and how it can be helpful for your research methodology, like research idea building, you please take a pause and think, if you want to make glyoxylate or succinate, then what you will do? Okay, tell me. Uh, Hope so you find it. If you want to make a succinate, what we can do, either we will delete this enzyme, MLS enzyme, or I will delete the conversion of the uh, isocetrate lyase is important. We cannot delete this, or we'll increase this and we'll delete MLS. Then what you will found? Then succinate and glyoxylate will be formed more, and it will not convert it into malate. Importantly, you understood. Other thing is that what we can do, why we need a succinate, because succinate is the intermediate in the several, like TCA cycle, it is intermediate, it is intermediate in the anapleuritic side pathways, and it is going to give you the immediate products. Like if you give the succinate, it will give you the immediate malate, or it will give you the immediate fumarate, and it will give you the immediate instant energy kind of thing we can understand, which is important. Like NADH, it will give, or FADH, it will give very soon. So these are the research paper which is done by the different scientists and the different people. What they did, if you can see this, that they took a strain which have a mutation in the ADHE and LDHA and ACKPTA and ICLR PHL 14 PH413. So these are the mutant. Okay, these are ADH, ALDH. What is this? These are the sequence of the gene which is going to synthesize a particular enzyme. Like citrate lyase is synthesized by the small ICLR. Okay, so if you mutate this, then it will be, what will be the effect? So if you will mutate this E. coli and if you will give a substrate as a food, glucose, then it will give what? Succinate. Okay. But if you don't want a succinate, then you have to mutate different thing. As you can see here, in E. coli, they mutated almost same, but they mutated extra MAEA B gene, and these all genes are mutated for the particular synthesis of the what? Mallet. They use the sgRNA and set to, like they did the knockdown also. They given the substrate glucose and they give the mallet. You must have known about the GABA. GABA is a neurotransmitter. It will help in the in cell signaling, importantly in the immune response also. So if you want more GABA for production, you will give a substrate glucose, and then you will mutate LAC1, GABT, and then PTA. So I cannot explain all the pathways at the moment now, but the concept is that if you will mutate certain enzymes, suppose if you will mutated the MLS, the MLS will not, the glyoxylate cannot form into the malate, and isocetrate lyase is converted the isocetrate into the succinate. Okay, so these are the important thing of the understanding what you want and which gene you have to mutate, and what product you want, what substrate is it is required. Okay, so this is the important thing. Like butanol is mostly used in the commercial things. So for making a butanol or hydroxypropionate, these are the acetate are the substrate, and these are the mutation which you have to do in the different organism like Pseudomonas or the E. coli. And hopes you understand this thing. If you want anything more, please let me know, and I will try to help you over there. So does bacteria have a glyoxysome? That is a question because we seen E. coli and these all some bacteria present here. And if does it have a glyoxysome? That is my question. Please do comment and make these classes interactive so I can respond these answers in the next videos. Thanks for watching. We'll meet in the another video. Jai Hind.